If you want to validate a drug target, where do you even begin? There are so many places to start and so many paths to take. Do you try and isolate and solve a three-dimensional structure of your target? Do you try and figure out what the mechanisms of action are? Do you try and figure out where your target is expressed? What if you don't have a disease model? How do you know which pharmacological agents to test? Looking to validate a drug target can appear to be a monolithic task with so many aspects to answer. In this video, we're going to have a look at some grounded strategies that can help demonstrate how studies are designed to validate a drug target. So let's dive right in. First things first, search the literature thoroughly. The first thing you want to do is understand what has already been done in the field and to identify a gap in the current knowledge on the topic. If you're unsure where to start, or even if you have an idea of where to start, the first thing you should always do is to consult published and peer-reviewed articles. Chances are that whatever you are thinking has probably already been done, so there's no point repeating the same experiments and coming to the same conclusions. This principle holds true for any research and study design. So say you've done your due diligence and you've explored all the literature in the topic, and you might find yourself in the following situations. One, there is an existing phenotypic disease model, for example, a mouse model that displays diabetic symptoms. So here we have a representation of a disease model, but little to no understanding of the underlying molecular causes. Or alternatively, there's a specific target involved in a pathophysiological mechanism, for example, a virus that is propagated by a self-engineered protease. In this case, we have a very specific biomolecule that appears to be very important to the viral life cycle, but there is minimal connection to whether targeting it will impact the disease progression. What might you do with this information? What's the next step to consider? So we can approach this in two ways, forward and reverse approaches. The distinction between these is their starting point. In a forward approach, you have a phenotypic disease model, and you're working towards finding a specific target or gene that, when modulated, addresses the symptoms or modifies the disease state. In a reverse approach, the opposite is true. You have a good understanding of a potential target, and you're trying to determine whether there is a clear link to treat the disease. These approaches represent a conceptual mapping of linking one end to the other. So ultimately, it doesn't matter what your starting point is, you should still want to fill in all the gaps regardless. So if we relate this to the examples introduced before, in the case where we have a mouse model that displays diabetic symptoms, our starting point is we have a phenotypic disease model, and we're trying to identify a protein or a gene that targets and addresses the disease state. In other words, this example represents the starting point for a forward approach. When we look at the other example that was described, a protease in a viral mechanism, our starting point is a known target, and we want to ensure it's linked to the disease state and safe to target. This would represent a reverse approach. Simply put, a forward approach seeks to find and link a target and molecular mechanisms to an established disease model, and a reverse approach seeks to link a known potential target to a disease phenotype. It's also important to keep in mind that different diseases have different mechanisms, and the experiments and techniques employed in a target validation strategy should be well considered and context specific. So how do we go about this? How do we design experiments to test these hypotheses, or confirm or reject the relationship between diseases and their investigated targets? So to address this, there are a broad set of tools, but in this video, we'll focus on genetic and pharmacological approaches. In essence, we're trying to investigate these disease models and targets by modulating its activity, and we do this through interfering with the gene expression or treating with pharmacological agents. Let's have a look at and contextualize these approaches using crossovers between our forward and reverse and genetic and pharmacological examples. If we put forward and reverse approaches on this end and pharmacological and genetic approaches here, we can look at how they might be used in each situation. So let's start by grounding ourselves in the problem. So our diabetic mouse model is a phenotypic model, meaning we observe a disease state first and then work forward to uncover the biology driving it. That's the starting point for our forward approaches. On the other hand, our viral protease is already encoded in the viral genome and is a known molecular target. That makes it a classic example of a reverse approach. where we start with a defined target and work backward to understand its impact on the disease. From these two starting points, we can imagine 4 experimental scenarios to explore target validity. In scenario 1, we can use a genetic approach in mice by modifying the genome to control the expression of a specific gene to see whether the changes in gene expression influence diabetes pathogenesis. In scenario 2, we can also use genetic approaches to knock down the translation of the viral protease by infecting airway epithelial cells and using small interfering RNA to reduce protease expression and observe whether this improves cell survival. In scenario 3, we can take a pharmacological approach in the diabetic mouse model, treating animals with a diverse set of drug-like compounds to see whether the disease-related symptoms improve. And then finally, in scenario 4, we can evaluate the viral protease using biochemical binding assays, comparing how well fragments and hit compounds bind to the viral protease versus similar endogenous proteases to assess selectivity. So let's zoom in and take a closer look at each of these scenarios in a bit more detail. So in scenario 1, we generate a transgenic mouse model where we can selectively switch off a gene that we believe contributes to diabetes progression. So in this scenario, we already have evidence suggesting that modulation of this gene influences disease progression. We also know that when mice are fed a high-fat diet, they develop metabolic symptoms that resemble those seen in human diabetes. So by knocking out this gene, we want to determine whether improved outcomes are directly caused by reduced function and presence of that target. At the same time, as this is a receptor expressed in the normal genome, we can assess how important this gene is for normal physiological function. If suppressing the gene improves disease symptoms at a whole organism level, that strongly supports the biological relevance of this target. We also gain early insights into safety by observing whether the loss of the gene causes adverse effects elsewhere in the organism. Nonetheless, this approach doesn't tell us much about the molecular mechanisms at play. We still don't know which signaling pathways the target controls or how it contributes to disease at a cellular level. It also doesn't tell us about whether the target is druggable or whether similar proteins might cause selectivity issues. Additionally, this is a low throughput and resource intensive experiment, so negative results can limit future directions. In scenario 2, we infect healthy epithelial cells with the virus and measure how well those cells survive. So we know the viral genome has been sequenced and that we can design siRNA to specifically block transcription of the viral protease. By silencing the protease, we want to determine how critical it is for viral replication and whether inducing its expression improves cell survival. If cells survive better when the protease is silenced, that provides a strong evidence that the protease has a biologically relevant function for viral propagation and represents a valid antiviral target. However, siRNA is highly selective, which means this experiment tells us very little about whether the target is druggable and if we can achieve the same specificity in modulating the function through pharmacological means. Furthermore, this is still a simplified system, so we don't capture interactions across tissues or gain insight into systemic responses to these changes. Additionally, this type of target can be prone to mutations, and we may not be certain on if the therapeutic we develop will still be relevant in the near future. Now, let's consider scenario 3, where we shift from genetics to pharmacology. Here we treat the diabetic mouse model with a range of drug-like, structurally diverse compounds and monitor disease-relevant endpoints, such as glucose levels, insulin sensitivity, or body weight. We already know that this mouse model reliably displays diabetic-like symptoms, providing a robust system for screening therapeutics. We want to see whether the disease symptoms can be alleviated through a pharmacological means, regardless of whether we fully understand their molecular targets at this stage. This helps us identify a chemical starting point for future development. If a compound improves physiological outcomes, it supports the relevance of the underlying biological pathways being modulated, ultimately building a strong connection to biological relevance of the impacted target. Furthermore, it supports the druggability of the target as we observe a direct, efficacious outcome by treating the disease model with a pharmacological agent. Additionally, we gain insight into the use of key biomarkers in the disease state, enhancing the assayability and translation between animal models and human patients. However, because this is a phenotypic approach, we lack clarity on the molecular mechanisms of drug target activity, where multiple targets may contribute synergistically to the observed effects. Additionally, as we don't know the specific target the drug is acting on, it's difficult to ensure safety in patients with comorbidities, where they might require the use of multiple medicines. Finally, in scenario 4, we focus on biochemical binding assays. Here we test whether small molecules bind selectively to the viral protease compared to endogenous proteases found in host cells. We assume the viral protease has been structurally characterized or purified, and that the closely related endogenous proteases are available for comparison. So in this scenario, we want to determine whether the compounds can selectively bind to the viral protease compared to normally expressed host proteases. Demonstrating selective binding strengthens confidence that the protease is a druggable target that small molecules can bind to. Additionally, comparing the binding against endogenous proteases show an indication for the ability to selectively target the viral protease pharmacologically, supporting the safety profile of this target. We also demonstrate a clear ability to perform high throughput screening and a proficient assayability of the target for further studies. However, binding alone doesn't guarantee functional inhibition or antiviral efficacy, as it doesn't guarantee that the hit compounds are able to inhibit the protease activity, ultimately giving us limited insight into the aspects of druggability. We also have an understanding on biological relevance, as this experiment is removed from complex physiological models that are indicative of efficacy and the ability to treat the disease state. So these scenarios highlight some of the general approaches and considerations to validating drug targets using forward and reverse logic, as well as genetic and pharmacological tools and techniques. There are many more different methods and experiments that can be used to answer these key criteria for a valid target. Nonetheless, the principles of forward and reverse approaches are consistently applied across all of them. The general logic for a research and investigative approach is to understand what is known about the scenario and identify a gap, then design an experiment that addresses or provides information on the gap, and finally acknowledge the limitations and need for further investigations, or to move on to the next stage of discovery and development. Validating a drug target can be a head-spinning task, but the best place to start is always the literature. Reviewing published work shows you what's already been done, inspires you for future experiments, and addresses the gaps that need further attention. From there, focusing on identifying what's missing and what experimental designs might be able to address the gap. Maybe there's an established disease model or a known potential target like a key protease. Each starting point helps you choose the next steps to investigate, but don't think of it as a linear path. Consider it more as a puzzle. The more you piece together, the clearer it becomes. To test these ideas, there are a number of approaches. We discussed genetic and pharmacological in forward and reverse contexts, but in this modern age, there are a multitude of tools that go beyond altering gene expression or using compounds to see how systems respond. But the most important thing is to be critical about the experimental results and what they really tell you. No matter the method, the goal is the same. Connect the evidence, address the gaps, strengthen the justification for selecting a drug target, and create a comprehensive picture of the drug target you want to pursue in the clinic.